Hi class and welcome to the final chapter. This will be chapter 28 in your textbooks and in Connect. The reproductive system we'll cover just briefly. There's only about 20 slides. The majority is just focusing on the anatomy of the system that you'll cover in lab as well. We first need to cover some lingo and understand the difference between gonads and gametes. Both systems have primary sex organs called gonads. These are the ovaries in the females and the testes in the males. And the gonads produce sex cells called gametes. And the gametes in the female are the oocytes or egg, and the sperm in the male. Both systems have a series of ducts to transport the gametes from the gonads to the site of fertilization where the gametes will fuse. And that's where we're going to focus on this series of ducts that lead to the transportation of those gametes. At puberty, the external sex characteristics become prominent, reproductive organs become functional, gametes mature, and the gonads produce sex hormones. Puberty will always begin when your hypothalamus increases release of gonadotropin releasing hormone, that's GnRH, and GnRH will stimulate the anterior pituitary to secrete follicle stimulating hormone and luteinizing hormone, which will stimulate the gonads then to start secreting gametes. So this is just a good kind of comparison between females and males and the common functions of the two. So I'll let you guys read through that on your own, just basic anatomy and the homologogs, the similarities between the two. So the testes and the seminiferous tubules, the seminiferous tubules will be located within the testes. They're a series of tightly wound tubules where sperm are formed. When sperm are formed, they will travel to the epididymis, which is kind of like this worm-like structure which sits on top of the testes, where the sperm will be stored until they're completely mature and until they exit out of the ductus deferens, which is the tube that will take the sperm out. The spermatic cord contains the ductus deferens and all of the blood vessels and nerves. The testes, the seminiferous tubules contain two types of cells, the sustentacular cells, which are non-dividing support cells that assist with sperm development. These cells are connected to each other by tight junctions that form the blood-testes barrier. A population of dividing germ cells continuously produce sperm beginning at puberty, and the spaces surrounding the seminiferous tubules are called interstitial spaces, where interstitial cells reside and produce hormones called androgens. Spermatogenesis, which is the development of sperm, occurs in the seminiferous tubules. It takes about 2 to 2.5 months to complete. For each sperm cell, spermatogonia lie near the base of each tubule, and they will divide through mitosis to produce new spermatogonia. Some spermatogonia will develop into primary spermatocytes, which are cells committed to becoming sperm. The hormonal regulation of sperm production, it occurs within the seminiferous tubules beginning at puberty. It uses the same set of hormones that females use during the ovarian cycle. So your hypothalamus releases GnRH, which is released every 60 to 90 minutes in the male. It targets the anterior pituitary gland to release FSH and LH. In the male, follicle stimulating hormone stimulates the sustentacular cells to promote the formation of sperm cells, and luteinizing hormone activates the interstitial cells to release testosterone. This has many male sexual characteristic functions, as well as causing the spermatogenic cells to develop into sperm. Negative feedback is required to shut down the pituitary gland, so increasing testosterone levels inhibit the pituitary gland from releasing FSH and LH when the levels are appropriate. And once the levels fall, the testosterone levels, the inhibition is lost and testosterone levels will increase again. This is the difference between mitosis and meiosis that we'll talk about briefly. Mitosis happens in 4 stages. It happens in somatic cells. The purpose is cellular proliferation, and it produces 2 identical daughter cells. The chromosome number remains the same and genetic variation doesn't change. Meiosis, however, is the specialized type of cell division that reduces the chromosome number by half, creating 4 haploid cells, which are each genetically unique or distinct from the parent cell. And meiosis happens in all the germ cells, so the egg and sperm cells. And the purpose is for cellular reproduction so that the cells produced from meiosis, the sperm and egg, with half the number of chromosomes will be able to combine through fertilization to produce a cell with the complete set of chromosomes. So the primary spermatocytes are diploid. They'll have 46 chromosomes, but they will undergo meiosis 1 to form 2 haploid secondary spermatocytes, which have 23 chromosomes. Secondary spermatocytes complete meiosis 2 to form 4 spermatids, which are also haploid. And spermiogenesis is the process in which the spermatid will differentiate into a mature spermatoza. So this is just the stage of spermatogenesis. Spermatogonium divide by mitosis. They contain 46 chromosomes. The primary spermatocyte contains 46 chromosomes, and it will undergo meiosis and develop 2 secondary spermatocytes. The secondary spermatocytes contain 23 chromosomes. Each one undergoes meiosis 2 to form 2 spermatids for a total of 4. And there will be early spermatids, eventually progressing into late spermatids, which will eventually mature into sperm cells. Spermiogenesis is the last part of spermatogenesis, where the spermatid transforms into the sperm itself. The acrosome formation is a sac that covers the head of the sperm with enzymes necessary to penetrate the egg. Mitochondrial reproduction forms a tight coiled spiral around the neck of the sperm cell to give it ATP for energy to stimulate the flagellum movement. And flagellum formation, the flagellum is formed for movement of the sperm to try to reach the egg. Oogenesis then is the production of the egg, the ova. Unlike the male, it begins during fetal development. So females that are developing in the womb will have their number of eggs already. It starts as an oogonium containing 46 sets of chromosomes, and it divides to form millions of copies. Some of these will develop further into primary oocytes, and they'll start meiosis division in fetal development, but they won't finish until puberty. And at birth, about 400,000 to 4 million primary oocytes will remain in each ovary. By puberty, about 40,000 remain, and 400 to 500 of those will be released from the ovary. The finishing of meiosis will now start during puberty, regulated by hormones, and this will occur monthly. So these are the stages of follicle maturation in the ovary. The primordial follicle is the primary oocyte. There's about 1.5 million at birth. The primary follicle is the maturing primordial follicle. It will contain the zona pellucida, which is the protective layer. The secondary follicle has multiple layers of the granular cells around the zona pellucida. And then the tertiary follicle has fluid-filled region called the antrum. And it also has a corona radiata, which is the innermost layer of granular cells. So same type of hormonal control. Your hypothalamus will release GnRH. It's not released in pulses like the male, but it'll fluctuate in females. GnRH will cause the release of FSH and LH. FSH will stimulate the follicles in the ovary by increasing follicular fluid and size, and luteinizing hormone causes estrogen release from the follicles. The initial levels of estrogen cause mostly a buildup of luteinizing hormone waiting for release later, and then further development of the tertiary follicle will produce more estrogen, and this will signal the release of the built-up LH in a big surge. This 1 to 3 day surge of luteinizing hormone triggers the formation of the secondary oocyte in the tertiary follicle. Luteinizing hormone triggers the rupture of the tertiary follicle starting ovulation and the release of the secondary oocyte. This will then drop the estrogen levels. Luteinizing hormone surge also changes the damaged follicle into corpus luteum, which will function as a temporary endocrine gland releasing estrogen, progesterone, and inhibin. And these 3 hormones will cause the decrease of GnRH, FSH, and LH. The corpus luteum will shrink and become the corpus albicans without fertilization, and this causes the hypothalamus to start releasing GnRH again and start kind of this cycle over. Fertilization, if the ova is fertilized, which always occurs in the uterine tube and becomes implanted in the uterus, it will release human chorionic gonadotropin, HCG. which is the hormone that is detected on pregnancy tests. This will cause the corpus luteum to continue secreting progesterone for about 3 months, and this will build up the uterine lining um, to help with the implantation and kind of development of the fetus. After 3 months, the corpus luteum will have regressed into the corpus albicans. So this just takes you through the stages of follicle development within an ovary, eventually until we get the ovulated oocyte being released. This just is a look moving kind of A through E of all the kind of development of those oocytes. And this is another look of oogenesis. Again, a good slide to just pause the video to record and read through. The wall of the uterus, the uterine wall has three layers: the perimetrium, the myometrium, and the endometrium. The myometrium is the thick muscular wall. The endometrium has a basal and functional layer. The functional layer is what will change thickness during the uterine cycle and is shed as menses or as the menstruation if fertilization and implantation does not occur. The uterine cycle refers to the changes in the endometrium as influenced by estrogen and progesterone. There's three phases. Menstrual phases are the menstrual bleeding. Reduced blood flow to the endometrium causes tissues to deteriorate and tissue is sloughed off. Then the proliferative phase is the development of the endometrium's functional layer. And then the secretory phase is increased vascularization and development of uterine glands due to high progesterone levels. This is controlled by estrogen and progesterone, and the purpose is to prepare the uterus for the implantation of the embryo if it happens. And if no implantation occurs, both the ovarian and uterine cycles begin once again with the release of GnRH. And this just takes you through the different levels of FSH, LH, what the follicles are looking like, levels of estrogen and progesterone, and what the lining wall of the endometrium looks like through a normal typical cycle. So this is a comparison of the ovarian and uterine cycles to kind of look at what happens in each phase: the ovarian cycle phase, so what's happening with the ovaries and the egg, and what happens with the uterus as it prepares for a possible implantation. Here's a good chart that summarizes the influence of hormones on ovarian and uterine cycles. So again, pause the video, read through this that takes a summary of the different hormones, the primary source of the hormone, and its effects on the ovarian and uterine cycles. And that's the end of this slide. You guys will learn a lot about the anatomy in lab. And thanks for listening.